Assalamu alaikum. My name is Ayesha Gull from Guwahati University of Science and Technology, Department of Microbiology. Today I present the topic study of the growing incidence of resistance to antibiotic and pathogenic bacteria associated with patient of general surgery. Now these are contents like introduction, objective, material methods, results, conclusion, and ideal references. First of all, we discuss introduction. Surgery or surgical procedures are medical practices that are used to treat or heal injuries, wounds, pathological condition of patients that are normally not treated by simple procedures. So it also included other condition of patients like cutting or surgery of tissue, insertion of implant or tissue grafting, etc. So healthcare associated infections are seen as a global public health threat. Pakistan, one of the developing countries, also facing the same challenges, and there are numerous reports highlighting drug resistance. Additionally, other factors like emergency procedures, longer duration of surgery, and hygienic conditions and different environmental factors can contribute in dissemination of resistant clones. Expansion of surveillance, prevention of infections, and antimicrobial stewardship programs are some of the steps that can be considered preventing or minimize the spread of resistant strains in surgical patients. So these are our objective. Number first, to characterize antibiotic resistant bacteria isolated from patient surgical site infection. Number second, to check antibiotic resistant profile of resistant bacterial isolate. Now we discuss material and methods. Number first is sample collection. In sample collection, clinical samples collected from surgical infection site of pathogens admitted or visited DHQ hospital KDA Kahar. Number second is culturing of sample. In this, each swab plated on different culture media including blood agar, McConaughey agar, mannitol salt agar, or nutrient agar. For isolation of susceptible bacteria, all plates incubated at 37 degrees centigrade for 24 hours. Now, identification of isolate and identification means like gram staining and different other biochemical tests like oxidase, catalase, coagulase, etc. Now, the next one is the antibiotic susceptibility test. In the different antibiotics are used against different bacteria like gram positive and like gram negative. The next one is DNA extraction of selected strain and resistant strain. In this, resistant bacterial strains inoculated in albumidia and well incubated for 24 hours. After getting proper colony, it's subject for DNA extraction using phenol coliform method. The last one is antibiotic resistant gene characterization and material method. So in this, on initial susceptible screening of resistant pathogens, resistant strain like multi-drug resistant or extreme drug resistant pathogen. Okay, pathogen was further screened for its resistant gene profile for this most prevalent resistant gene like TEM, SHB, or CTX screen. Now the next one is result. Frequency of bacterial isolate. Total 100 wound samples are obtained from DHQ hospital KDA cohort. The percentage of Pseudomonas aeruginosa is 35%, Staph aureus 40%, E. coli 15%, Bacillus cereus 5%, Proteus species 5%. All of these were found to be the predominant agents isolates from the wound sample. Next is identification of isolate. Identification involves the following steps like examination with naked eye to observe the colony morphology including shape, size, surface structure, age, color or opacity. All the isolates were identified based on their biochemical tests. Gram stains were prepared from the colonies to notice the shape, size, arrangement, and staining reaction. Now in this slide, the picture shows the gram positive have purplish bluish color and gram negative have pink or reddish color. Now the next one is biochemical test. In biochemical test, different tests are like catalase, oxidase, indole, etc. So the Catalyst test is positive all gram-negative bacteria like E. coli, Pseudomonas species, and Proteus species, while oxidase is positive for Pseudomonas or negative for both E. coli or Proteus species. Indole test is positive for E. coli or negative for Pseudomonas or Proteus. Citrate test is negative for E. coli, while positive for Pseudomonas aeruginosa and Proteus species. Urease is negative for both E. coli and Pseudomonas, while positive for Proteus species. So, motility E. coli, Pseudomonas, and Proteus are motile. Now we discuss antibiotic sensitivity tests. So antibiotic sensitivity of all bacteria isolate were tested against different antibiotic like vancomycin, ampicillin, oxacillin, imipenem, chloramphenicol, kanamycin, erythromycin, cholesterol sulfite, levofloxacin, antibiotic this, etc. In this slide show the most effective antibiotic against all the strain is levofloxacin because it shows the highest zone for all of them. The least effective antibiotic for E. coli and Staph aureus is cholesterol sulfite because it shows the lowest zone, while for Pseudomonas aeruginosa, the least effective antibiotic is chloramphenicol. In this vancomycin, oxacillin, erythromycin, and ampicillin are not effective against any of the strain because they show no zone. In DNA extraction of selected resistant strain, in this resistant bacterial strain inoculated in algae media and incubated for 18 to 24 hours after getting proper colony is subjected for DNA extraction using phenol chloroform method. In this picture showed it. So for DNA extraction, first of all, we extract DNA from sample, then run on the gel for the confirmation. When bands show, so it's mean DNA is properly extracted. Okay. So in this figure clearly show the bands. Another hand, we also run leader for positive control. These are references. Thanks to all of you for concentration and for precious time.